You know, every single living thing, from a massive redwood tree all the way down to you and me, is built from one master plan, DNA. It's the blueprint of life. But what's amazing is that this entire grand code relies on a partnership between two very different molecular families. And today, we're going to dive into that story.
Okay, so right at the heart of our genetic code are these two families of molecules. And you can see the main difference right away, can't you? It's their size. On one hand, you've got the purines, these bigger molecules with a double-ring structure. And on the other, you have the pyrimidines, their smaller single-ringed partners. Now, this size difference isn't just some random little detail. Trust me, it's the key to absolutely everything.
So before we get into how they work together, let's get properly introduced. It's time to meet the individual members of these two crucial families.
First up, the purines. These are the big guys, the ones defined by that two-ring structure. And in this family, there are two key members you gotta know. Adenine, which we call A, and guanine, or G. And you'll find both of these in DNA and RNA.
And here's their partner family, the pyrimidines. See? Much smaller, with just that one single ring. This family has three main players. First is cytosine, or C, which, like the purines, is in both DNA and RNA. But then you have thymine, T, which is only found in DNA. And its cousin, uracil, or U, which steps in to take thymine's place, but only in RNA.
All right, now that we've met them, the plot thickens. It turns out these two families have completely different life stories, from how they're built inside our cells to what's left of them when they're finally broken down.
And check out this contrast in how they're made. The purines are kind of built right on site, assembled piece by piece directly onto a sugar molecule that's already there. But the pyrimidines, they're more like prefabricated. The ring gets built first, all by itself, and then the whole thing is attached to the sugar. I mean, how cool is that? Two totally different manufacturing processes.
And the way they break down is just as different. When our body is done with the pyrimidines, they break down into stuff that's easy to get rid of, like urea. But the purines, their breakdown produces something called uric acid. And that can have some very real-world consequences.
For example, if your body makes too much uric acid or just can't clear it out fast enough, it can build up and form these sharp little crystals in your joints. This causes a really painful condition called gout, and it's a direct result of how this one specific family of molecules, the purines, gets broken down.
Okay, so this brings us to the main event. We've seen these two families are different in size, in how they're made, and how they're broken down. So now, let's see how they have to come together to build the ladder of life following some very, very strict rules.
Which leads to a really fundamental question, right? Why? Why does it absolutely have to be that a big purine always pairs up with a small pyrimidine?
And the answer is structural perfection. It's a principle we call complementarity, and it ensures that the DNA double helix keeps a perfectly uniform width all the way down. Think about it, it's like a ladder. If you tried to pair two big purines, the rung would bulge out. Two small pyrimidines, it would pinch in. But one big one and one small one, that makes every single rung the exact same length. Perfect.
But wait, it gets even more specific. The pairing isn't random. Adenine, the A, will only pair with thymine, T, and they connect with two hydrogen bonds. Guanine, or G, on the other hand, only pairs with cytosine, C, and they form a much stronger connection, with three hydrogen bonds.
So at the end of the day, it all comes down to this simple little fact. 3 is greater than 2. That one extra bond in the guanine-cytosine pair makes a world of difference. It literally changes everything about how our DNA behaves.
So let's really dig into that. What are the massive consequences of that one extra bond? Why does this simple difference matter so much?
This visual says it all. That GC pair, with its 3 bonds, is like a door with an extra deadbolt lock. It's way more stable and takes a lot more energy, like higher heat, to pull it apart. And that has huge implications for everything from how our genes are controlled to the basic stability of our entire genome.
So let's do a quick recap of the whole story. We've got the structural difference, 2-ring purines versus 1-ring pyrimidines. We met the family members, A, G, C, T, and U. We saw how one family is built on-site, the other is prefabricated. They break down into totally different things. And most importantly, we learn their strict pairing rules that hold our very essence together.
So, in the end, this isn't just a tale of two families. It's a story of a perfect partnership.
From their different sizes to the exact number of bonds they form, these differences aren't mistakes. They're the very features that create a stable, replicable, and nearly perfect information system. It's a molecular dance that is, quite literally, the foundation of life itself.